Hello, welcome everybody to the Fall 2022 Cancer Care Delivery Committee. It's so great to see everybody here in person. And for those of you in the audience, we also have another 44 people on Zoom. So super excited about this hybrid meeting. So welcome to Chicago. And just in terms of housekeeping, we're gonna be, because we have so many, actually 52, because we have so many people on Zoom. For the people that are on Zoom, I hope that you can hear me, but I'm gonna ask you to type in any questions you have. I'm gonna be monitoring the questions that come in. If you feel like you really wanna state your question, just raise your hand and I'll find an opportune time for you to ask your question, and you can unmute. So that way, I think we'll make it as interactive as we possibly can. But don't hesitate if you're out there in the Zoom world to feel like you can participate. With that being said, we have a packed agenda, and so I will hand it over to Scott.
Great. Hi, everybody, and welcome back to Chicago. I mean, I'm also thrilled to see everybody in 3 dimensions as opposed to on screens, so thanks for that. So I am really excited to present our first speaker. This is Dr. Vita Henderson. Dr. Henderson works with me at the Fred Hutch, where she's an assistant professor in public health sciences. She is a pharmacist and a behavioral science expert. And her presentation today is on development and testing of a culturally responsive decision aid intervention to promote diversity in clinical trial participation. And as you can tell from that title, could there be a better fit for this committee? I don't think so. So Dr. Henderson, we'll turn it over to you. Come on up.
Morning, everybody. Thank you for inviting me to be here today. Let's see. So as Scott just mentioned, today I'll be talking about work focused on facilitating decision-making about clinical trial participation among Black breast cancer patients. The name of the study is called the For Me Study, and that stands for Fostering Opportunities in Research Through Messaging and Education. It's a grant that's funded by the American Cancer Society, and it's a 4-year project period, and we started this year. So this is our amazing research team. I've been at Fred Hutch for about a year, and before that I was at University of Illinois in Chicago. So we're definitely a group of collaborators from Fred Hutch, from UIC, and from Mount Sinai, also here in Chicago. For those who aren't familiar with the University of Illinois Health and Hospital System, it is a large academic center, but it's also affiliated with a network of federally qualified health centers. So in a lot of ways, even though it's a large academic medical center, it also serves as a safety net hospital. And in that realm, so does Mount Sinai. So these 2 hospitals serve a lot of people on the south and west sides of Chicago that are disproportionately impacted by cancer disparities. We're working with a group of amazing community partners that we've been working with over the years and have really strong relationships with. And this is very much a project that incorporates approaches of community-based participatory research. So as we all know, Black women die at a 40% higher rate than white women from breast cancer. But they're grossly underrepresented in cancer clinical trials. And so we need to really develop interventions that look at multi-level factors to increase equity in clinical trial participation and inclusion and representation. So the primary objective of this decision aid narrative video, it's really to help increase awareness about clinical trials and to help Black women who have breast cancer really think about, is participating in this clinical trial right for me? So we're gonna start with I'm a qualitative researcher, so we're starting with doing interviews with Black breast cancer patients, just to understand their experiences, their values, their needs when it comes to clinical trial participation. We're also going to talk to oncologists, clinical trial staff, and community advocates, and to understand their perspectives and experiences as well. And then we'll take all of that qualitative data from interviews, focus groups, story circles, and we'll develop a narrative intervention. And when I say narrative, it means like there's a storyline in it. Some of the work we've done, it includes live actors, animation. So we kind of listen to what people tell us, what they want it to look like, what they want the content to be. And we try to develop a script and a storyboard and images to create this video or decision aid intervention. And we really hope that by using all of these methods that we end up with a product that's authentic and relevant that will really resonate with the audiences that we're using this for. So this is a representation of kind of our overall approach to how our team develops decision aids. It's adapted from a previous model by Whitman. So as I just mentioned, we start by understanding the user and different stakeholders. And then we take that information and we develop the tool and we do multiple iterations to refine it with those same stakeholders through focus groups, additional interviews until we get to our final product. And then we pilot test it in the appropriate setting, whether that's a clinical setting or a community setting. Our approach differs a little bit from Whitman in that we include aspects of cultural tailoring and targeting. We aim to have a low cost intervention that can be easily disseminated. So we incorporate aspects of health information technology. And as I said, we also incorporate aspects of storytelling because we know that stories are really an effective way to elicit emotional response. And with health communication research, we know when we can trigger those emotions and transport people, they're more likely to engage in health behavior change. So Matthew Kruder is a well-known health communication researcher, really prolific, and he developed 5 strategies for enhancing cultural appropriateness in interventions. And just really quickly, some of the things that you want to do whenever you're doing culturally tailored or targeted interventions, you want to make sure that you use images that look like the people that you're working with, your group of focus. You want to use evidence that's representative of that target group. Obviously, you want to present things in the dominant languages of that group. And one thing that we found is really effective is to draw from the experiences that we hear and incorporate that into the narrative, and really making sure that the intervention incorporates cultural aspects and social aspects that people are dealing with and their values. So we have 3 project aims. I already talked about aim 1 and 2. We'll be doing those qualitative methods with those different stakeholders to understand barriers, motivating factors. What are some of the things that would make you want to participate in a clinical trial? And then to use all of that information to then develop the narrative intervention. And then for aim 3, we're gonna determine the acceptability of the intervention and conduct a pilot study in the UIC oncology clinic. So this project is guided by 2 theoretical frameworks. The first is the integrative model of behavioral prediction, and that asserts that the primary driver of behavior change is a person's intention to engage in that behavior. But we know that even if a person has the intention to engage in a behavior, there may be external factors that either help or hinder their ability to actually do so. And then we also are guided by the model for analysis of population health and health disparities, which was developed by Dick Warnke, who recently passed away. So I refer to that as the Warnke model. But it's an ecological framework that basically asserts that the causal fundamental predictors of health behaviors and health outcomes, they span the range of biological pathways, individual factors, community factors, and social and policy factors. So we're looking at all of these constructs in developing this intervention. And basically this asserts, like, even though participating in a clinical trial seems to be an individual decision, there's a lot of external factors that go into play into why somebody would decide or not decide, or if they're able to participate. So this is an example. As I said, we're doing interviews, focus groups, story circles. Story circles are similar to focus groups in that instead of having a formal, semi-structured focus group guide, you just introduce a prompt and then you just let people talk. And you do get different layers of information when you use different approaches. And to get at those ecological levels that I just talked about with the Wernicke model, for example, we have different interview guides for each cohort. So we have patients, like I said, providers, and community advocates. So for an institutional level factor for patients, for example, we're asking, have you ever experienced times when you felt that you were being mistreated, unheard, or felt like you were getting less quality care while getting health care? Would you tell me about that? Has that experience, if at all, has it impacted the way you feel about healthcare providers or the healthcare system? So these are just kind of examples of what I mean when we're trying to get at those different layers, ecological factors. So this is our study design overview. I already talked about aims 1 and 2. For aim 3A, we're conducting an acceptability pilot study. This again will be conducted in the UIC Oncology Clinic. We'll conduct a pre-survey. We'll have 30 breast cancer patients watch the video, and then we'll take a post-survey. And that post-survey comprises a validated acceptability scale, just to understand their feelings about the actual intervention. For aim 3B, we'll be doing a clinical trial uptake pilot test. And what we're doing for that, so at UIC in the oncology clinic, as part of standard of care, when someone's diagnosed with breast cancer, they sit with a breast cancer clinical nurse navigator, and they have an educational consult. So as part of that education, we're gonna incorporate the video into that. So what we're gonna look at is previous 12 months before we started implementing the intervention, what was the clinical trial uptake, and we'll compare that from when we start for another 12 months when we start the intervention, and compare and see if clinical trial participation rates among Black breast cancer patients increased in this particular clinic. This is just a table showing the number of participants that we'll be recruiting. So it'll be a total of 120 participants, unique participants in all, participating in various aspects of the intervention development. I talked about that we're using CBPR, community-based participatory research approaches. Not gonna go through all of this, but one example is for the story circles that I talked about, we're actually working with cancer survivors who are part of our community partners from Sisters Working It Out and Equal Hope. And we're actually training them to conduct the story circles because they're survivors, and we just think that it would be a more impactful way to have that conversation with other Black breast cancer patients. So we're training them to do that data collection, to participate in the analysis, help us interpret results, and for dissemination. We were also fortunate to apply for a grant from the V Foundation this summer, and we were awarded this grant, and we're using somewhat similar methodology, but the whole goal of this is to develop a public service announcement for general Black communities, general audiences, just to talk about what clinical trials are. We thought about using like business marketing strategies to really go beyond academic and clinical walls, to really disseminate this public health research and this message to a broader audience. So we're working with marketing professionals who we developed an interview guide. We're gonna be conducting focus group guide, and they're gonna give their input on that from a marketing perspective. They're gonna be the ones conducting those focus groups, and we have a professional recruitment agency helping us recruit, and we're working with our video marketing company that we often work with. And for the For Me study, we started recruitment last week, and we already have 11 breast cancer patients who have signed up to do interviews. And we started recruiting for the V Foundation study this week, and we already have some people who are going to participate. And for the V Foundation study, we are looking to do focus groups with just general audience, people in the general public from Black communities, and also social support networks of cancer patients. So progress to date, we've completed all of our regulatory approvals. We started recruitment. We'll probably start the interviews actually before the end of this month, probably next week. We've presented our development strategy at several meetings. We've done some trainings, and we're continuing to do team building and partnership engagement. And that's it. I will try my best to answer any questions that anybody has.
Any questions about these efforts?
I'm Barbara Segarra, a patient advocate for cancer care delivery. I love the project, especially because you're involving the patients and other things. So before you presented the sponsorship you have now by the B Foundation for the general public, that is one of the things that I think that we have to start at. And I wanted to ask you if in those focus group, if they were asked if they know what a clinical trial is, because my argument is that when you get sick, you're trying to understand what cancer is, and then trying to understand what a clinical trial is even worse, right? So if people can talk about it, and I think after COVID, I had the opportunity to talk about FDA and what trials are, and I think we're in the great moment for the general public to understand. So that was, I wanted to know if from the focus group you got that, and I love the project. Thank you.
Yeah, I think that might be question number one. Like, do you know what a clinical trial is? How would you define it? And then, of course, we offer a definition, but I think with a lot of the work that I do, a lot of the stuff that I do is health education. And a lot of it starts with like the basic nugget. Even when I'm doing like mammography, advocacy, it's amazing how many people still think, oh, you don't get a mammogram until you have a symptom. So that's why these the educational tools really have to kind of break things down. And I think that's a lot. We just have this assumption that people know what clinical trials are, but to really understand what they think it is really important. And then to kind of target our messaging around what we find out. So.
This is Marie Bakaitis, co-chair of the Palliative and End of Life Committee, and also qualitative researcher. So I had actually a couple of questions about getting through the process of regulatory and approval with a CVPR study. Bravo for doing it, first of all. But I wanted to know if you could give us some helpful tips about that. And then the second question relates to the protocol. You have navigators that work specifically with women with breast cancer, and I suspect most organizations do or don't. Were they in any way involved in the development of the video?
So we haven't developed the video yet. So that's what all of this work is leading up to. And actually, that's a good point that you're making, because we seem like we were involving as many people as we can, but I don't think we actually did plan to talk to the clinical breast nurse navigators. So that's a great point that I'll bring back. So thank you for that. In terms of IRB, yes, we got the approval for this grant in January. We just got IRB approval. Plus, it's 2 different institutions, so there's all this back and forth. So it's a lot. I think one of my pet peeves is, especially when we're working with community-based organizations we keep saying we want them involved in research, but we don't make it easy. And so you have to get people who want to go through this CITI hours of training, and it really is taxing. So luckily we got 2 people who actually did it and wanted to do it, so that's great. But I think there needs to be better mechanisms to get community advocates and partners involved to actually really be involved in our research.
I think that's kind of my point, because we have a couple of studies that have community advisory groups and others that have like real community advisors to the study.
Mhm.
And we have, the city training is a huge barrier.
Yes,
I
absolutely.
mean, you're assuming a whole lot about people being able to, and that's the people that we're trying to attract and to inform us are people who don't necessarily have the resources and education and background and everything to do the city training. And
Yes.
so, I mean, it's really an IRB educational issue or institutional regulatory issue. If we are truly going to do community-based research, we really have to look very carefully at that requirement.
Yes, absolutely agree.
It's an extremely important point in general as we think about expanding, our pool of patients and trying to increase the diversity of our patients, like to figure out how to resolve those kinds of structural barriers, to allow sites we were talking about this earlier today, maybe partnerships, so that we can have sites that don't have the clinical trials infrastructure, but have the patients that we really want to be engaging in research, so that we can figure out how to solve those really deep structural barriers to trial participation. There are a couple.
Yes, absolutely. Yes, for every encounter, they're getting paid. Yes, every interview, every focus group. We're even offering transportation. If they want to do it in person, we're paying for their transportation to come on site, parking stickers, all of that.
So there are a couple comments that came in through Zoom, and one is that is about some of the work that's happening in the digital engagement committee, focused on some similar things like creating videos. I don't know, Don, if you want to mention that work to maybe make that connection.
So I actually had two comments for you. The first is that the Digital Engagement Committee, Jonathan Summers is one of our patient advocates, and he got funding from the Hope Foundation to look at videos. And it's remarkable just about how they're composed and how much time people actually spent on the videos and what might be the sweet spot based on who you're trying to reach, how you can target that group. So it might be worthwhile for you to talk to him, because I think it's really exciting. And DE, we were thinking about how we can transform what we've learned into a project in cancer care and delivery. The second is that what I've learned in some of the work I do in community outreach and engagement is that not everybody's on the same platform. So understanding where that community meets is really, really critical before you develop your PSA, and that some communities are going to be more responsive to clinical trials that are actually linked to a disease rather than to something about clinical trials in general. Mm-hmm. So, oftentimes the important hook might be breast cancer, or it might be this, rather than let's learn about clinical trials.
Yeah, and I appreciate that, especially with the PSA. We don't really know what the platform will be. It may be a TikTok video, who knows? We want to really understand where people get their information. And we also want it to be engaging. I don't want to say fun, but something interesting for them to look at, to where they would want to share it with their network. So thank you. I appreciate that comment.
Yeah, I mean, along the same lines, the Hope Foundation just funded a seed grant through funding we had for clinical trials, improving clinical trials, specifically looking at creating content for AYA patients specifically, using a variety of different types of social media platforms like TikTok. And so it might be good to connect with, we can help you make those connections to sort of get on the, sort of pool all of these resources together, and then figure out how we can use all of the work that you're doing to then be able to apply it in these kinds of settings.
Thank you so much.
Thank you.
All right, I'm up next. So This is going to be kind of our swan song for Tracer 1415 CD. We, as you know, have finished that trial and have got our preliminary results. Both our primary outcome and the secondary outcome are going to be published either this month or next online in JAMA Network Open and JCO. So we wanted to give everyone who's rode with us alongside that study a preview of what you're going to see in those manuscripts. So I think everybody in this room knows this. We did a study to investigate this issue of using primary prophylactic colony stimulating factors for patients receiving myelosuppressive chemotherapy. We know the factors are expensive. Historically, they have been used not at all according to guidelines. And kind of wrapped within that is this issue of using colony stimulating factors for people receiving intermediate risk chemotherapy, not high or low risk chemotherapy, that the benefits and costs of using it in that setting are uncertain. So this is 1415 CD. So our primary objective was to see whether the guideline-informed standing order entry could improve colony stimulating factor prescribing and then reduce the incidence of what we want it to do, which is reduced febrile neutropenia. This was conducted in the NCORP CCD sites, and we basically broke it into whether you had an existing order entry system for colony stimulating factors. If you did, you were put in a cohort study. If you did not, you were eligible for randomization into usual care, which is just keep doing what you're doing, or the order entry intervention, which was that we would upgrade your EMR to have an automated system for putting in colony stimulating factor orders according to NCCN guidelines into your system. We also had some paper-based systems, and we did it in paper when we had to. Within that intervention arm, we had a second intervention arm for individuals who were receiving intermediate risk therapy. They were sub-randomized either into the system auto-populating for colony stimulating factor orders, or in the other hand, not auto-populating, and the provider would get an alert that CSF was not recommended. In all of these interventions, the provider was allowed to override the orders if they felt that that was appropriate. And so we ended up with shooting for 90 patients per site, and the idea was to detect an FN risk reduction from 15% to 7.5% in the intermediate arm. So these are the results of the study. And just want to shout out that this was, I think the largest recruitment we've ever had for a pragmatic randomized trial or a trial in cancer care delivery. We had 3,600 patients, 2,800 into the RCT and 700 into the cohort arm. The average age was 58. The majority were breast cancer patients, but we had good representation among lung cancer and colorectal cancer patients as well. Now, what happened with adherence? So to our surprise, what we found is that there was very high adherence to colony stimulating factor use, both in the high risk arm and in the low risk arm. In other words, for patients who were supposed to get colony stimulating factor because they were getting high risk chemotherapy, we found in the usual care arm that 95% of those individuals did indeed get colony stimulating factors. And also to our surprise, in the intervention arm, and remember that was the arm where it was auto-populated, you were getting it, actually a lower proportion got colony stimulating factors. So that meant the physicians had to override the orders to some degree. Now that was not a statistically significant difference, but it was surprising. We thought if anything, at least there would be more prescribing in the intervention arm. Similarly, in the low risk arm, we found very low rates overall of use of colony stimulating factors. And in fact, in the intervention arm, again, where the order entry system said, do not prescribe, we're not auto-populating, there was actually a slightly higher proportion that overrode those orders and prescribed colony-stimulating factors. And if you look over in the cohort arm, over on the right there, you'll see similarly that there was fairly high use of CSF in the high-risk group and relatively low prescribing, 8.3% in the low-risk group, although that was a little higher than what we saw in the trial arm. Now the FN rates with that relatively high adherence ended up being rather low. So for the high-risk group, the FN rates overall was 5.7 in the intervention arm and 4.3 in usual care. Again, that was not statistically significant in terms of its difference. And the FN rates were very low, as expected in the low-risk group, and no statistically significant difference there as well. Now let's talk about that sub-study, that secondary randomization. Here's where things, I think, got a little bit more interesting. So we recall that we were, in one arm, everyone was auto-populated with orders in one group, and then in clinics that were randomized to receive no orders, no CSF, there was an alert. This, in contrast to the primary study, there was an impact of the order entry system on adherence. In fact, the odds ratio was highly significant in favor of the order entry system influencing use in this group. So among the 524 patients in this group, 37% who were in the prescribe arm received colony-stimulating factors. Now, it wasn't 100%, which perhaps you might think would happen with auto-population. So there was some overriding, but you contrast that with 9% in the no CSF arm, that was highly significant. Also interesting was the rate of febrile neutropenia in these groups. Basically, it was very low, much lower than one might have predicted based on the guidelines and what they estimate the risk is, which is kind of between 10 and 20%. In both of these arms, the rate of febrile neutropenia was about 4%, and it was not significantly different between arms. So a much lower rate of FN in this particular type of chemotherapy category, for which we had really no evidence about what that rate was. So what did we learn from this study? Now I'm going to kind of just do a few takeaways. This study, I will say, I think has given some kind of boost to those who want to do cluster randomized trials, and I think that's great. I think that's an important tool we have for cancer care delivery research. But I got to say, this was hard. When you look at the NCORP site, they say there's 1,000 clinics. It's like, oh, we've got all the clinics we need. Of course, that doesn't account for the subsection of those that are CCD clinics. And then among those who are CCD, finding practices that actually want to participate. We struggled to get the 45 we needed to be a part of this trial. The other surprise, as I mentioned, was that adherence to colony-stimulating factor guidelines was much better than predicted. And there were multiple studies, including studies that our group at the Hutch had done, that had shown very poor adherence to colony-stimulating factor prescribing prior to the date of this study. And to be honest, we still don't exactly know why this happened. Perhaps the practices just improved their adherence over time, and their generics came online, and there was a lot of more aggressive work on insurance companies with pathways companies that were really pushing, trying to push down or to push the use of these drugs to more appropriate levels. But it's also possible that there was a Hawthorne effect, that people knew they were being observed, and as a result, they adhered more closely. So that's something we're still looking into. The other thing we learned is that, man, there's a lot of kind of chaos in the world of oncology practice. First of all, just getting this intervention into 7 separate electronic health records and 3 practices that had to do paper was hard. but it was the norm to expect that practices were going to change fundamentally. Their ownership was going to change, their leadership was going to change. We were going to lose the CRA champions who were at the sites. And so our team just had to be in constant contact to be ready for those things and to get in there and to try to help, maintain the integrity of the study over time. And I think this is a lesson for all of us in cancer care delivery, that just expect things are going to change over a study that goes on for years. All that said, I think this was a big win for us at SWOG and for CCD specifically. We did get 38 NCARB sites and 7 minority underserved NCARB sites to participate. That was across 25 states and territories, including Puerto Rico, who did this through a hurricane and continued to recruit. I'm still amazed at that. And we were successful. We made it through, even with COVID. We finished strong as COVID was ramping up. All the intervention sites were able to modify their chemotherapy order systems. So if we want to do this again, we have now proven that you can do it. And that really was the site champions, the folks that said, this is a priority, we're going to do it. And I just want to thank all of you who were part of that. We had very low dropout, and we met our accrual goals, as I said, during pandemics and hurricanes and other disasters. So it was really amazing. We also had very good, unusually good enrollment of Hispanic patients into this, and that was exceeded what we've seen for other SWOG network trials. And I think this study is going to be influential. On the clinical ground, as I mentioned, we found very low rates of febrile neutropenia in this intermediate group that has not been shown before. And I really think that's going to be practice-changing evidence. And after these studies are published, I'm sure the guideline groups are going to look at that data and hopefully revise their recommendations for the use of colony-stimulating factors accordingly. And if we do find lower use of CSF in that group, that's going to save our system hundreds of millions of dollars over the coming years with no harm to patients. In fact, benefit to patients, because they won't be on the hook for out-of-pocket payments for those drugs. As I ran this trial, I've just kind of kept track of what was going in the world of, modifying EHRs to direct physicians and all these assisted entry systems. There's a lot of enthusiasm out there in the tech community and elsewhere for doing that. I think this trial is going to give people pause before they invest all the time and resources into modifying systems on the assumption that it's going to change practice. We've shown that you can't assume that. And I do think that's an important lesson, because standing up things like this is expensive. And we don't want to waste people's time if we aren't going to change practice to the point where we want it to change. For the trial community, I thought this was a big win. For those who are interested in pragmatic trials, and a lot of folks outside of this room still don't know what a pragmatic trial is, but we've shown that you can conduct one, it can be successful, and it can be done in a way that has direct relevance to those who are in the field, who are taking care of patients. I also just have to give a shout out to the patient advocates, using them from the very beginning as trial advisors, as co-investigators in this study. I think made this study successful. Their input was so critical. They had so many insights that we took and modified our proposal, both at the start and during the trial, as we learned, that allowed us to be successful. And any study I do in the future, they're going to be right there in the study helping us design it, because that input is just so critical. And I think for the NCARBs, we've now expanded the idea or the ability to do what's possible in a trial. And so we can do these trials at a large scale in this network. We can do something that's relatively complicated, like modifying order entry systems. And I think that landscape has changed now, and I think Tracer I hope had an influence to encourage all of you here and in the other groups to think big when they're talking about modifying trials to modify practice behavior. I want to acknowledge my co-chairs, the biostatistical group at SWOG, our external stakeholder advisory group, who included folks internal to SWOG and external to SWOG who were so helpful. And of course, my research staff, who just, again, they made this happen. And I'm so grateful for all the hard work and enthusiasm they put into this study. Thank you.
So we'll take questions. Anybody that's online, feel free to type them in.
Rick.
Hi, I'm Rick Bangs. I'm the chair of the Patient Advocate Committee. I'm curious about the overrides. Has there been any analysis done as to whether that was clustered in certain practitioners or whether it was kind of uniform? Because my suspicion is there's some change management involved here, and you've got a set of practitioners that are probably responsible for the overrides.
It's a great question, and we've started to look in the variability and adherence across clinics, and frankly, it's huge, and Dawn has been kind of taking the lead on that. But I don't think we've looked specifically at the provider level yet to see if there were some, good actors and bad actors, if you will, in terms of overriding.
Yeah, I think Bill is going to be presenting some of that variability next when we talk a little bit about the design issues. So you can stay on the edge of your seat to see some of those numbers.
Great presentation, Scott. I'm just thinking clinically, when I put in a prescription for a colony stimulating factor, it's a generation of a bunch of emails to the billing people, which one is covered, do they need to come back into the clinic, or can they self-inject? And so I'm wondering if there were, if these order entries sort of interfaced with the back end of pharmacy billing and authorization, and if there might have been differences in the clinics in terms of how they handled after the order was placed, getting access.
Yeah, we've actually there wasn't, we weren't specifically collecting that data, but we have started asking clinics about, because of this unexpected outcome, what was going on. And I actually suspect at the back end, there was a lot of stuff going on in terms of influencing the orders, like just as you described.
And I think it's an important point in terms of collecting data going forward on these trials, and we've certainly built that in to our newer, generation of trials, of really making sure up front we're collecting data and asking clinics about their behavior so we can better understand the changes over time that are happening, so we can answer these kinds of questions at the end of the trial.
But yeah, to that point, we didn't collect through the entire chain. That starts with the order on the Epic, and then it goes to the pharmacist, and then the pharmacist has to check what the patient's insurance is, and the insurance has to say yes or no. That's just stuff we're going to have to, we learned that we have to track as part of studies like this where we're affecting cancer care delivery, because the insurance rules, the practice rules are all in there, and we need to track them.
So I would just like to express, our thanks. This was one of the very first intervention studies that was done in cancer care delivery across the entire network. And I hope all of you take home the comment that you made, Scott, about this being practice changing. That is really what should motivate you doing your work back at home, and why these studies really matter to our patients and to us as organizations and health systems. So thank you to the study team who did a huge amount of work, I know, and to all of you at the sites who conducted this study, because it really was a heavy lift and has made a difference.
Yeah, if you can inform, practices, providers by saying what they're doing that's not actually helpful for de-escalation, it's a challenging thing. And to have level 1 evidence that we can guide, at least for first line therapy that's intermediate risk, that they're not getting a benefit from giving this, is really important.
thank you. Guys, you're thanking the wrong person.
And I was part of Carol as well. And I think this group really involved the patients, and we felt part of the team all the time, and they were able to, we were able to be freely express our ideas, and we see the change. So that was really powerful for all of us that participated. And I remember one of the meetings that it said, oh, they were starting to report recruiting, and in Puerto Rico, we were in the middle of the Hurricane Maria, and the whole island was without power for many, many months. And Scott said, even Puerto Rico had the first recruited patient before anybody else. And when Magna called me about a new trial that she wanted to contact people in Puerto Rico, when I called them, they're begging to have more trials in Spanish for them to participate. So I encourage, we have the resources. I'm going to talk a little bit later what we can do, but we're out there, and the people in Puerto Rico are very committed, and it's not a public service, but it is. And they participate, and they stay in the trial. So thank you, Scott, for including us and for a great trial. And I did take the stimulating hormones, so that when I was a patient, and the pain in the joints is terrible. So if you can spare that with this to the patient, it's gonna be great.
Yeah, I think it's a win-win, especially that intermediate risk group. Maybe the companies lose, but that's okay.
I mean, I think it's also worth thinking, because there was a 6-fold difference, but in that intermediate where we really didn't know between, the sites that had the standard order entry and the sites that got the alert not to use it. And we should really tap into that for more questions, in scenarios where we really don't know, because it was a very effective strategy when there's a lot of uncertainty. I think when there's less uncertainty, harder to move the needle. But there are many, many situations in care delivery where there's a lot of uncertainty. And we can take this, what we've learned from this, and apply it to other situations. So it's worth thinking about other scenarios where this may be effective.
Yeah, I mean, a place where there's clinical equipoise, where cancer care delivery is part and parcel of it, that's where I think we want to go. So if you guys were thinking about it, if you guys have ideas in that area, we want to hear, because we'd like to kind of keep the momentum going.
I'm not a patient advocate, but I do want to say something. I think it would be fabulous if once you had your paper, you could provide a synopsis that was IRB approved, that we could send to our participants that we enrolled. And unfortunately, some of mine have died, but I would love to be able to show, what their participation.
Yeah, that was actually that's actually part of our plan, is to come up with dissemination to patients. And we'll have Barbara and others help us figure out how to boil that down.
Great. So Bill is next.
Yeah, Bill's going to give us the statistical version of our study.
So I was asked to talk about pragmatic trials by Scott, and in the process of getting ready for the talk, I saw Dawn's presentation to ASCO and said, this is far better than anything I was doing. So with her permission, I borrowed several of her slides at the beginning of the presentation. I'm actually not going to talk that much about 1415, but a little bit at the end. But let's see.
Oh, you have to press that.
Oh,
right. There you go.
Okay. So again, this is from Don's ASCO Quality of Care Symposium. And typically we think about traditional randomized controls trials as being the gold standard for evaluating health interventions, but they're also designed to maximize internal validity, and often at the cost of generalizability. And that's really to reduce heterogeneity in the patient population so that you have a clean comparison. It just may not apply to very many people. So pragmatic RCTs are designed with a conscious effort at generating evidence that's going to have greater external validity by making the research question as similar as possible to the question you really want to ask, the question faced by clinical decision makers. And they're designed to test effectiveness rather than testing efficacy of an intervention. And sometimes you see this described as testing in the real world. I actually don't like that phrase very much. Our clinical trials have real world patients undergoing lots of testing, lots of treatment. And so I think it's a little pejorative to say clinical trials are not real world. But I do agree with the idea that we want to have results that are generalizable to a larger population. So they're intended to improve practice and policy, and they can take place in different settings where everyday care happens in community clinics, hospitals, health systems, as evidenced by 1415 CD, include diverse patient populations. 1415 CD intentionally enrolled several minority sites into the population. They were also randomized. It was a stratified randomization for minority sites as well. We have collaborating providers and organizations that are integral partners to the trial and gain practical evidence on how to actually improve patient health and satisfaction. So we traditionally present this as a dichotomy between a treatment trial or traditional randomized trial and a pragmatic clinical trial. And where the goal of the treatment trial is really, does the treatment work? And the goal of the pragmatic trial is really to inform practice and inform clinical and policy decisions about, do you offer this drug to the community? So it's a different question that you're testing. Oftentimes in a traditional test, you may have placebo. You would never do that in a pragmatic trial. You would never have a placebo condition in a pragmatic trial. Participants may be highly selected in a traditional trial and less so in a pragmatic trial because you relax the eligibility requirements. And I think one of the most important aspects of a pragmatic trial is really the data collection outside routine clinical care. Traditional treatment trial, there are lots of case report forms. You make them come for extra visits. You make them adhere to the randomized arm, or you try to get them to adhere to the randomized arm. In a pragmatic trial, the true pragmatic trial wouldn't have a case report form. You would get the data from the electronic health record system. And all the data would be the same data that a typical provider would see for a patient not enrolling in a clinical trial. So you would randomize clinics, get the data from the healthcare system about whether they progressed, for example, and that would be the trial. That's what back when we called it a simple pragmatic trial. We don't do that any longer because we realized how difficult these trials really are to do. So we've dropped that adjective. And we end up with results that are probably more useful in everyday practice, including clinical decision-making. Now, we often present these as two alternatives, and that's not really the case. They're actually on a continuum, and there's a scale that measures that continuum from an explanatory trial, why does this treatment work, versus a pragmatic trial. And there's a scale called a PRECISE-2 scale that is actually measures on 9 different domains and uses Likert scaling from 1 to 5 to decide how pragmatic is the trial that you are designing. And this was first developed as part of the consort working group, the figure that everyone has to put in their treatment trial, saying what happened to the patients. And now we all are required to do this. Well, it's been proposed that this instrument for measuring whether it's pragmatic or not also be incorporated into clinical trial planning, and that you have to fill out this form. I'm actually hoping that doesn't happen. because it's more complicated than it looks. But there is a website now for this form, and looking at these 9 domains, it's actually been, so Dalray and et al. Actually looked at trials that were labeled as pragmatic in the title, but they were studying drugs. And of the 89 trials that claimed that they were being pragmatic, 36 of them were not in fact pragmatic, either because they had placebo controls or they were at a single institution. Sometimes pragmatic is a code word for not controlled. And that's an unfortunate use of the term, because people will apply the term pragmatic with the implication that it's somehow going to be more generalizable because of that. Netta and Johnson actually looked at this tool that is suggested, and actually showed that it's difficult to use in practice, and it has very low inter-rater agreement for raters looking at the same trial, whether it's pragmatic or not. Now, there is a question of pragmatic versus implementation science. Those two are complementary. They actually grew up in totally isolated from each other. Pragmatic trials were invented in the early 2000s. Implementation science also started in the early 2000s. They basically coexisted in the same space with never talking to each other for about 10 years. And now there's some communication between these two areas. Implementation science is implementing changes or testing the implementation of changes that you know have been successful elsewhere. And they actually, this article by Poston, who's clearly an implementation scientist, was arguing that pragmatic trials are not really often very pragmatic, and that may not be as generalizable as we think, because it depends on the clinicians chosen for the trials, it depends on the practice settings chosen for the trials. And the implementation science often argues that you have to do a multi-level approach. You have to attack at the healthcare system level, the provider system, and patients all at the same time to test your implementation. You can't just attack one of those levels. But he goes on to say, well, how are you going to actually test implementation science? And then he says, well, basically you have to run a cluster randomized trial at one level to actually test some of the elements of implementation science. So cluster randomized trials are used when it's not practical to randomize patients, so you randomize providers or practices or clinics. Sometimes patients would be randomized, but one is worried about contamination during distorting the trial. I would argue that's not a reason to do a cluster randomized trial. It has very few advantages compared to the disadvantages with respect to power, and I'll show that in a moment. You're better off living with contamination and the effect on power, which is to lower power somewhat, versus doing a cluster randomized trial because you're worried about contamination. So the statistical methods we use for analyzing patient outcomes in cluster randomized studies have to account for the correlation of patients within the same randomized clinic or randomized unit. And so statistically, we have one of two ways of doing that. We add a random effect for a clinic in a mixed model. That's probably the more common approach. Or we use a GEE model, or the generalized estimating equation model, adjusts for the variability in the background, or just for this correlation in the background for the lack of independence within patients. They give you fairly comparable results. In 1415, we actually did both. The mixed model was the primary analysis, and the GE model was a sensitivity analysis, but the results were the same. And why does this matter? Well, the correlation between patients, let's say it's actually between 0 and 0.1, so a very low correlation level between patients in the same clinic. If you do a sample size calculation, that's going to adjust for that correlation. So let me just give you an example of that. Let's say we had 40 patients per clinic, and we want to get 80% power, effect size is 0.25, we have a continuous outcome. How many clinics of size 40 would we need? Depends on what you think the interclass correlation is. So at 0, which is essentially a randomized trial of individuals, you would actually need about 6 clinics of 40 patients each in each arm to do the trial. If the correlation between patients is only 0.02 between, so it seems pretty low, but it would require almost twice as many patients to do that cluster randomized trial as opposed to an individual randomization of patients, just because of that small correlation. And the reason is because with 40 patients per clinic and correlation between any 2 individuals in that clinic of 0.02, there are 780 pairwise combinations of the 2 patients. So that's a whole lot of correlation going on, which is why it builds up so fast and has such a damaging effect on the overall trial power. So I only showed this going up to 0.1 because it becomes impossible to actually do the trial pretty much after that point. You will get to, you need an infinite number of clinics if the correlation is too high. So how does this relate back to 1415? This is my conclusion. We had a wide variance in the number of patients per site. So we planned to have an equal number of patients. We knew it wasn't going to happen, but in fact, it was a wider variation than we thought. That we were trying to get 90 patients per site, I think. We set a cap of 200 patients per site. So the problem with this is in an individual randomized trial, every patient counts the same. In a cluster randomized trial, that is not the same. That if a site has a large number of patients, the addition of one more patient to that site is smaller, has a smaller effect on the trial than the addition of a patient at a clinic with small numbers. So we had capped it at 200, and in fact, it's hard to discourage your really good sites that are really good at accruing to say stop accruing because it's not going to be helpful to the trial. So you do have to manage the number of patients within a clinic that are getting randomized. So when you design these trials, you actually want to maximize the number of clinics you have or number of providers, and you want to minimize the number of patients per clinic because of this interclass correlation. So in terms of design of 1415, we assume that the interclass correlation was 0.02. That's a fairly common assumption in these cluster randomized trials, somewhere in that order. And it comes from patient data that suggests that might be reasonable. And we correct for patient factors, so some of the correlation goes away because of that. The actual ICC for assessing prescribings of colonnade stimulating factor was 0.2 in this trial. So that means we didn't have power to actually find anything because of that high correlation. And I thought about it afterwards and realized, oh, it's obvious why that is the case. It's the physician is controlling the prescribing. We usually think of the ICC as being a patient level variable, but the physician is controlling the prescribing. So therefore, the correlation within patients of that physician is extremely high. In fact, it was like 0.39 for in this trial. So it's very difficult if you're going to look at physician behavior, then the ICC is going to be much larger than you would expect. For febrile neutropenia, which is a patient outcome, there was no correlation at all within patients. It was entirely did not depend on clinic whatsoever. And that, so it was estimated to be 0. So that would have meant no effect on power. Unfortunately, the results weren't in the right direction, so we couldn't actually find significance. But so I'll stop there, but it certainly was a great effort, I think, in 14-15 CD. I'm amazed at how well they did. I'm sorry the results didn't turn out to be in a direction we think, but it turned it out to be more interesting in some ways. And I learned a lot about doing cluster randomized trials in the process. Thank you.
I mean, just to, extend that, what we found was that just in terms of variability, there were entire sites that were randomized to the intervention that had no use, and other sites that were randomized to the intervention that had like 70% use. And so, people, it says a lot about physician behavior, right? Because to not do what you're told to do, you have to make an effort. You have to override the system. And so our job in terms of changing people's beliefs when they really strongly believe something is a challenging one. But we should acknowledge that is, and it was different from practice to practice. So it's not just the physician, it's the environment they're in. So there's a question here wanting to know whether or not the practices that overrid worked in a capitated system, and that we really don't know. But to agree to participate in the study, the sites had to upfront state that they were willing to be in these randomized arms. So it wasn't being done to them. They had to agree to be part of the study.
And there were some sites who refused because they said, basically, we're not going to follow that. And that was okay. So they weren't part of the study.
Hi, I'm Kathy Crew from Columbia. In terms of doing these cluster randomized trials, like the main challenges are, trying to get that balanced accrual across all of the sites, then also how to keep the control sites engaged when they know that they're not getting the active intervention. So what were sort of some strategies that you adopted to try to address those?
So I think it's really a challenge, I think, depending on the intervention. And when we start to think about other studies and usual care, like thinking about that is really important, because in this particular instance, because it was a drug as opposed to behavior, there was less need for engagement. But if you were asking sites to, to participate in some other kind of behavioral intervention, it could be a lot more challenging in that way. Or if the patients knew that they weren't being part of something, they also may have less interest in coming back and filling out forms and things like that. So I'm not really sure what the right answer to that is, but it's a good one, especially depending on the intervention that you're administering.
Yeah, and I mean, it's not that dissimilar from telling a patient they're going to be in the control arm of a clinical trial. How do you convince them that that's a contribution? I think certainly for the intermediate risk group question that we embedded in this, I think physicians were okay with this idea that there had to be a group that didn't get CSF so that they could be compared with the group that did. And I think Dawn's point about how you bring clinics into these type of studies by saying this is, you're going to help answer a clinical question that will help you take better care of patients. CCDR in general, I'm not sure everyone totally believes that this is real science and what we're doing matters. So you do have to have site champions saying, look, we're applying the same science that we use for clinical trials, and we're trying to answer questions that you all are facing in your practice about how to take care of people. It's a harder story to convey than this new, PD-L1 drug might work in kidney cancer. But it is, so it's just on us to be a little, we have to spend a little more time. We have to find the clinic champions who believe in us. And that's what happened in Tracer. Those are the unsung heroes of Tracer, is the clinic champions who took this forward.
I just wanted to say too that I think Ari and the efforts that your team made to engage us as sites was super important. And I loved the newsletters. I loved when we had the meetings together to collaborate. And I do think that made a difference.
I can't even begin to express my gratitude for my team, Ari and Kate, in terms of what they did. A lot of that was their own idea, and they just, they took this to heart. They to make sure that all of you on the ground understood that we were there. We wanted to share information. I mean, I really think that's such a critical take home for any of you who want to do these CCD studies, cluster randomized or not, is to just keep the clinics, keep them informed. Just don't start and then leave. You've got to be there. And in some cases, they were calling clinics every week. How's it going? Any trouble? It really did make a big difference.
It takes a village.
Boy, yeah.
So with that, maybe we'll move on. Thank you so much.
Thanks, Bill. All right. I think next is Jens or Meghna? Meghna, yes, okay. Meghna's going to provide an update on 2108 CD. This is the comparison of educationally enhanced genetic tumor board intervention to usual practice of incorporating genomic test results into clinical care. Long time coming, but just great to see this one launched, I gotta say.
Thank you. Do we have the slides?
It's the green.
Oh,
Hit that green button.
there we go. Perfect. So what an excellent lead in with all of the Tracer discussions and cluster randomized trial. It was really helpful to have the experiences of Tracer as we designed this trial and thought of a lot of these issues with having a cluster randomized design and how are we going to engage with the sites. So the study officially activated August 22nd. We have 16 of our 18 recruitment centers randomized, and we're planning to finalize the selection of the remaining recruitment centers by the end of this month. And we're expecting the first genomic tumor board to be either the end of this month or early in November. And this is the schema for the trial. I know everyone in this room has probably seen it a million times, but again, the intervention is looking at an educationally enhanced genomic tumor board. We are having a kickoff meeting later today between 2:30 and 3:30. It's going to be in person and virtual. If there is anyone online that doesn't have the virtual link, feel free to send me or Jens an email. In person, it's going to be in the Wrigley room on the concourse level in the West Tower. And this is going to be in lieu of our monthly October 2022 monthly meeting. So very brief update. Again, we're going to go into a lot more detail and answer questions during our kickoff session. Please join that. This is all of our contact information. Again, as Dawn said, it takes a village. So we have a lot of people involved in this. We want to make sure everyone knows who the right people are to contact. But if there's any hesitation of who you should be reaching out to, you can always reach out to Jens and I, and we're happy to answer any questions. That's it. That was my last slide.
Meghna, I don't know, or Jens, maybe just go through a little bit about the information that's being collected in the intervention versus the usual care arms.
Sure. So again, it's a multi-level data collection. So we're collecting data at the recruitment center level, then at the physician level, and then at the patient level. So we try to be very mindful, again, trying to understand what the burdens will be on the patients, the physicians, and the recruitment centers as we go through this process. We're looking to enroll over 1,100 patients. So it's going to be a lot of patients enrolled to this study. At the patient level, there are no PROs, so there's nothing that's being collected collected specifically from the patient when they sign consent. They're being enrolled into study in order to allow us to look into their medical record and get information from their medical record over a 2-year follow-up period, with the most intensive data being collected that first 6 months, and then its vital status updates up through 2 years. At the physician level, we are, again, very mindful of the very busy clinic practices. We're using mostly rural and underserved community sites. And so these are very busy clinicians. So we're trying to minimize their burden as well. So at the baseline and at the end of the study, which is about 24 to 27 months, there are about 20-minute surveys that the physicians, the clinicians will be completing. And then for each patient that they enroll, there's a very quick 6-month survey, essentially trying to understand if they believe that their patient received evidence-based genome-informed therapy, which is our primary endpoint. Then at the recruitment center level, we're going to be collecting practice descriptions and trying to understand the geography and the kind of community that the practice treats, and then also trying to understand what is the use of genomic testing at each recruitment center to try and understand who are the high utilizers, who are not. So each month they'll be submitting how many genomic tests are being submitted at that recruitment center. In the background, we are also doing an implementation objective where we're trying to understand how practice evolves over the study period, both in the control arms as well as in the recruitment center arms. And so we'll be conducting interviews with physicians in both arms over the period at 3 time points, at pre-implementation, mid-implementation, and end of study, to see how does practice change over the course of this intervention period. Precision medicine is rapidly evolving, and within months we can see lots of changes. And so we're hoping that these interviews will allow us to collect, mixed methods, so both qualitative as well as quantitative data on how practices evolve with the intervention and without the intervention.
Any questions?
Jens, I don't know if you have anything to add. Jens is in the front row.
And just to remind everybody again, you're having a focused meeting today.
Today at 2:30.
In the Wrigley room.
Exactly, yeah. And we have, of the recruitment centers we're spanning a broad part of our country, from Hawaii to Puerto Rico. So we're really excited for all the recruitment centers that have joined us, and excited to have the last 2 join at the end of the month. Great, thank you.
Thanks so much. Next up is Dr. Veena Shankaran, who will be talking about credit, S1912CD.
The green button, okay. Great, okay. I just want to give everyone an update on the credit study. This is just a refresher on the schema. So this is, just to remind you all, a randomized study, randomizing patient and a spouse partner dyads to an intervention consisting of financial navigation and counseling by our community partners versus financial literacy training alone. The intervention will be conducted over 6 months. We're collecting credit data and survey data from participants over the course of a year. Our primary endpoint for the study is household financial hardship. And we're looking at a number of other caregiver and patient outcomes as well. So in terms of updates, we at the April meeting had a roundtable, and we definitely appreciate all the hard work all the sites are doing to find dyads. This is a hard study to enroll to. We have 46 enrolled as of October 10th. Our total accrual target is 536 dyads. So we have a ways to go, and we've been active for a little over a year. It's definitely been slower than we anticipated. One of the big, we got a lot of great feedback from all of you at the last meeting, and we've been working the last 6 months on making some changes to the protocol, which were just approved. So I wanted to go over those. So moving forward, starting right now, these changes are in effect and hopefully will help. So there are three big changes. I would say the most significant one was we heard that it is very hard to find married dyads. That was our previous eligibility requirement. Now we have changed. We still want to keep the partner caregivers involved, but we're not requiring that they're legally married. They simply need to be a significant other living within the same household as the patient. So my hope is that for people in long-term relationships or that are not married, this would be helpful to get those folks enrolled. And I think as we move forward, depending on how accrual goes, we can sort of readdress that and maybe make some changes in the future. But I'm hopeful this will help. Obviously, I'd love to hear from all of you about what you think. The other two changes were to allow for people with hematologic malignancies that were indolent over a long period of time that then progressed to more aggressive disease to enroll, so long as the aggressive disease presentation that's requiring systemic therapy happened within the 120 days around registration. And the other change was to allow people with secondary malignancies, so long as those were not diagnosed in the last 24 months, but not requiring that they not remain on active, that they not be on active therapy, such as, for example, women on hormonal therapy for breast cancer. So I'm hoping these changes will help. And we do have some other changes that we are discussing. These are not yet approved. And I just am kind of mentioning it as a heads up. We are planning to extend the eligibility window from 120 to 180 days from diagnosis, just to allow a little bit more wiggle room. And we've had a few instances of people with CNS tumors who don't technically have metastatic disease. And we are modifying the eligibility to allow these people to go on study. And so if we have time, I'd love to hear from you all about what the current and proposed changes, what your reaction is to those, and maybe if there are other changes that might be helpful.
So there's a question, Vina, about whether or not a parent or child could be considered part of the dyad.
No.
And what your thoughts are about why.
Yeah, no, I think that's a great point. I mean, fully acknowledging that some people either don't have caregivers or the caregivers they have are not significant others. Right now, we haven't made that change to allow for that. I mean, it could be something we think about in the future, but it's still significant others only.
There's another question about the filing joint taxes if they're not legally married. Is that a requirement still?
It's no longer a requirement.
Okay, so that's good.
Hi, Virginia Sun from City of Hope. I was wondering, did you hear about specific reasons why it was challenging to enroll caregivers? Was it because they don't accompany the patients to the clinic, or just challenging in terms of getting them, introducing them to the study and for informed consent purposes?
Yeah, I think that's part of it. I mean, it's hard because we have to consent both parties concurrently, so it's hard, especially if only one person is coming into the clinic. That was definitely an issue during COVID when there was a limit as to who could accompany people to clinic. But I think it's even more than that. I mean, I think it's the requirement that people are legally married. I mean, we heard from Barbara, you mentioned that you had a lot of couples in Puerto Rico at your site that they weren't legally married, but they would have been willing to participate. So I think it's a combination of things, yeah.
Can I ask how many people in the room have this open at their site? Can you raise your hands? Dr. Shankaran, would really love to hear from you, and so would I at NCI. What do you think of this amendment, and do you see this as making a change? Please share your thoughts.
So in terms of the couples, every time I present, and last year I was invited to help Vina in this roundtable that we had, I try to consult other patient advocates. And when I put the requirement that we had, it was like, people don't get married these days, don't do that. So it's mostly because I have two sons, and the commitment is buying the house. That's I think the closest to marriage that you're gonna get. So that came from that. And my question to you, from those 46 then interested in the Spanish speaking, have we been successful in that, in recruiting Spanish speaking couples?
Oh, good question. I don't know the breakdown actually, Barbara, but we can find, yeah, I'll look into that and maybe email you.
Hi, Krista Brown-Inglis from Hawaii Minority Underserved NCORE. I think it helps a little. I still think we have a lot of those single patients that, maybe older with advanced disease that have a lot of concerns about their finance that would be willing to enroll, but maybe their partner has already died and they live with another family member. That's what we see in our clinic. But I do think the, extended eligibility helps, and not having to be married helps a little, just to be honest. But I know you have to keep, because of the tax returns and things like that.
Yeah, thank you,
I mean,
Krista.
that same question came up on the chat in a slightly different way, which is that a lot of people's electronic medical record does not capture marital status very well. And so for these trials where the staff is trying to screen eligible patients through the EMR, not feeling confident, it's really challenging to reach out to people and have the first question be, are you married? So it poses a challenging, it's more challenging from a, where do you start in terms of talking to patients?
Yeah, absolutely. I mean, I'm hoping there's a little bit less judgment now with the changes, implying that if people are not married, that,
Right.
But I still think finding those people is going to be a challenge. And especially, you're not going to find whether people are living with a significant other in the EMR at all. So I think this is just an ongoing question about, how much we stick with this particular dyadic arrangement, but I think it's worthwhile to do it at least for some time and see what happens.
Another thought I had, and we should do this within our NCORE, is talking to the infusion nurses more than maybe the screening, if they can help us to identify patients. So doing more education again in the clinics versus just using our regular screening methods, or relying on the physicians or the advanced practice providers to, identify patients as well, because they're talking to the patients, they're in the clinic. I know this comes up, but maybe we just need to think about it again, because you have that big eligibility window.
Great suggestion.
Another question was about other languages other than English and Spanish.
I wish we had more, but right now it's just English and Spanish.
One comment in terms of, even if they're not randomized to the intervention, the educational material alone is very good. I was reviewing the one in Spanish, and I learned a lot just reading what was put together. So I think it's important also, they will benefit just because it shows you how to read the health insurance and different things that are very important. So just the educational piece alone is really good. So maybe, what material we're using to promote the trial, maybe we can revise and see, how appealing it is, or what else can we add to it.
So another question that came up that was seen as a barrier was patients having to give their social security number, and that talking about money in the Midwest is hard enough as it is. But this asking for the social security number is near impossible. I would say, I don't know if that's a Midwest issue per se, but we maybe just talking about how you got over that with the prospective study.
Yeah, I haven't heard that as a huge barrier until now. With this study, it did come up a lot with 1417 in the beginning. And, I guess I would say we had some materials on privacy, on protections around the credit data pool not affecting credit, and we do have that as well for this study. It's essential, unfortunately, for us to be able to get that in order to pull the credit data, which we need. So, I mean, I'm curious if that's been a barrier at other sites. It sounds like maybe so.
So Marie Bacaitis from University of Alabama at Birmingham. note the geography. It's next to impossible to get anyone to give us a social security number for anything, for any of our studies. We've had to get, because our incentives had previously required providing a social security number. The incentives were no longer an incentive to participate in any of our trials. And so we either had to, people, if they really were motivated, they would say, well, I just won't take the incentive. And then ultimately we got a waiver of requiring a social security number for trials that offered incentives. So, I mean, I think that unfortunately, and I know why you need it, and maybe, you know, there's a way to incentivize people to want to provide it for the greater good. But I think that, you know, and also you have people who are, maybe their immigrant status is an issue. And so, and maybe those are not people that you want in the trial. But I'm just saying that that this requirement, from my perspective in the South, is going to be a big disincentive for people enrolling. It's just the reality.
So there are a couple questions about how hard it is to recruit patients with stage 4 cancer right when they're diagnosed. But I think you had mentioned that you extended the windows to try to help provide a longer period of time so that maybe you don't need to bring this up on their first visit, but once they get settled a little bit, then that may be an easier time to bring that up. I don't know.
Yeah, it's not, that change hasn't been implemented yet, but it will be 180 days. And we'll send out, you know, a notice when that's updated.
Okay.
Well, more to think about.
Yeah, no, this is really helpful. Thanks to everyone, and please email me or the credit team with any other thoughts along these lines. It's really helpful. So you're saying at the high enrolling sites for 14-17. Great idea. Yeah, thanks.
Thank you.
Thank you. Now we're on to the Sacradino study, 1703.
Hi.
Press that.
This? The green?
Green.
Oh, thank you. Well, good morning. It's good to see everyone in person again. I know we spoke earlier about studies where there are areas of uncertainty, and I want to highlight 1703, which is looking at how we monitor patients with metastatic breast cancer to ensure that they're on effective treatment. But there really is a lot of uncertainty in how to best monitor patients with metastatic disease. There really is no data in the space, and this study's an opportunity to change practice because we are collecting data on how to potentially best monitor patients in this space. So I appreciate everyone's hard work thus far, but we do have a ways to go. Here's our schema. I know we've shown this a billion times throughout these meetings, but this is a two-step registration process. We are looking for patients who have elevated tumor markers at the time of metastatic breast cancer, And we do have a pretty large window to enroll these patients based on some of the changes we've made. And they do get randomized if one of the tumor markers has decreased. And there is compensation for the sites for registering both to step 1 and to step 2. So this is an easy way to get credit with this study. In terms of where our enrollment stands, you can see we do have a ways to go. As of last week, we've enrolled 366 patients to step 1, and we've randomized 236. We have made some progress, but there definitely is opportunity. So we really would like to implore all of you to open the study if you haven't, and to continue to offer this to your patients, as this really is an area where we don't know what the right answer is, and we're hoping that the study will help us. We've taken some steps to improve accrual. We've discussed having champions from other cooperative groups. We are now having regular webinars monthly, and may go to every other month, with CRAs and PIs to answer questions. We are allowing remote consent for all reasons, not just COVID. We have a brochure that staff can post in the rooms. We're just working on final approvals for this. We do have a social media toolkit that we will be using, and we do have plans to update the non-inferiority margin to hopefully reduce the sample size somewhat. But here is the draft of the patient brochure that will be coming soon, that's meant to tell patients why we're doing the study, why it's important, how they can participate, and I'm happy to answer any questions.
So out of curiosity, I mean, I have quite a few patients on this study, or we do at our site, and it's pretty easy sell. Patients are usually pretty willing to go on. So I'm always a little surprised that it's not accruing more quickly. And so I don't know if there are any sites that haven't opened it or haven't wanted to open it, or haven't had patients that want to go on it if they have it open, to just give us any insights into that.
It has kind of felt like a no-brainer to me, but that may just mean that I have no brain. But but this one seems so easy. Is it just that people are already doing this and they don't feel the need for a study, or?
Could it be at the patient's level in terms of insecurity, thinking that the scans is the things that are really going to tell them that they, are progressing, and they don't rely on the tumor marker, and how we can, encourage that? And I remember when you were doing that, if the physician feels they could go ahead and do the scan. So I think that explanation is very important, because it happens, when you, as a patient, when you finish your treatment, you're afraid. And it's something like, it keeps you like, okay, something is happening. So maybe thinking that they're not going to have the scan, they're afraid that then it is going to progress and nobody's going to notice. So maybe at the patient level, that could be something.
Yeah, and I think, that's a really good point. And I know in terms of the FAQs for how to talk about the study with patients, we really want to emphasize that there really is no right answer right now on how to monitor these patients. It's really entirely made up. So we could either do what we consider to be usual care, or we could be following with the more potentially personalized approach. But you're right, it's really important to emphasize that at any time for any patient, regardless of what arm they're on, if they have any symptoms or any need to have scans for a medical reason, that's absolutely allowed, even if their tumor markers are not rising. If there's concerning signs or symptoms, or if another doctor needs to get a scan for another reason, that is permitted.
So just one question here, which I think you can address. There was a concern about having all the different tumor markers, and some physicians not believing that they need all of them. So
Yes.
Maybe just clarify that.
Yes, so when we first opened the study, we required CEA, CA 15-3, and 2729, but based on feedback we've received, we do not require 15-3 and 2729. You can choose one of them, you just have to be consistent. If you'd like to get all three, that's okay, but we really are only requiring 2729 or 15-3 and CEA.
Yeah, so there's a choice on that. So that's been a change since the beginning.
Hi, I'm Stephanie Smith. I'm with Georgia NCORE. We've enrolled several patients to this trial. There have been patients whose tumor markers don't normalize or come down, so they cannot go on to the study. We've had patients just flat out decline, so to answer the questions why. At one of my satellite sites, I only have one coordinator out there. And she's a phenomenal coordinator, but she's told me that there's times when she's very busy with patients on treatment trials. She just does not have time to screen patients for cancer control and CCDR studies. So that's something that we're looking at internally, but I imagine that there's a lot of NCORE clinics that are set up like that. And so when there's a lot of patients needing to be prioritized to treatment trials, then these trials fall to the wayside, unfortunately.
Yeah, and I really appreciate that comment. One of the strategies we've talked about on our monthly calls are, because it could be labor intensive to screen all these patients, is maybe to try and get a report from the EHR for new prescriptions for CDK4/6 inhibitors, and that might be an easy way to potentially identify eligible patients. I don't know if that would work with EHR systems that you have in clinic or other people do, but we've found using Epic that we've been able to screen for this study and for potentially others.
Great. Thank you.
Thank you.
So we now have just some quick updates, I think, of the developing studies. Now, I don't know if Emily or Karen are online.
Can you hear me? This is Emily. I'm online.
Yes, we can
Great.
hear you, and we can see
Okay,
your slides.
wonderful. Thank you. And Karen and I regret that we couldn't join you in person today. So we have an update on the study looking at effectiveness of an oral anti-cancer agent rapid pro model, which is a model utilizing a patient-reported outcome tool focused on oral anti-cancer agents, or OAAs. So the next slide. We submitted this protocol this past year, and it was reviewed by a special emphasis panel. And we scored 38 percentile with that review. So to share with the group the comments related to significance and innovation, we feel are easily addressable, and we'll comment or bring some of these up. And then the approach scored slightly worse is 4 to 6, also addressable. And I think really overall these comments have been incredibly helpful to think about feasibility of rolling this out within sites and practices. So we have met and hope to submit again. And I wanted to thank everyone for all the help and support with this, particularly the individuals at the Base Stats and Data Management Center in Columbia. And of course to Dawn and Joe for their support. So next slide is the schema to share this. So we intend or hope to randomize on the patient level a total of almost 800 patients. We're looking at patients who are prescribed tyrosine kinase inhibitors. This is a bit more narrow than our original attempt, which was to address all oral anti-cancer agents. Practices will be able to take part in this trial as long as they don't currently utilize patient-reported outcomes regularly for the management of their OAA patients taking TKIs. We know that there are multiple different levels of support to this patient population, and our main focus here is how a standardized tool could be implemented for multiple patients with different drugs in a site, and how that can help enhance the care to that population. There are 3 aims with the study. The first one, really looking at the primary outcome of treatment failure and looking at time to treatment failure with these patients and reasons of discontinuation, such as progression, treatment toxicity, or death when using this model. The intervention itself is the combination of the rapid pro being completed by the patient, and based on those responses, the outreach to the clinic, so that the clinician in the clinic has proactive outreach to the patient in response to their rapid pro. So again, those aims for the outcomes were discussed. We also hope to look at what differences exist on either the participant or practice end that may make this more feasible to implement, as well as that may change what types of outcomes we see from patients. And then finally, we'll plan to interview practices based on facilitators and barriers of this protocol. So the next slide. We have 2 slides that really look at what is our current status and why is this unique. So what is unique about the Rapid Pro model compared to current programming? One aspect related to pros and the benefit in clinical practice, that is quite well known. What isn't known is how that can be integrated into everyday practice for patients receiving oral anti-cancer agents. The dosing, as in how often should they fill out or complete pros, is unknown. And can we have a standardized approach given that there are 80+ medications versus customizing for each of those? How do we work with staff and staff constraints in the current setting are all important factors that our model will look at. Another comment is really that OAAs can liken this cancer treatment to more chronic conditions compared to the traditional IV treatment model. And so another aspect that's measured with the Rapid Pro and that we hope to analyze is how with this programming and this intervention can practices really support patients in self-management. So the self-management and confidence in the ability to manage the side effects they receive with these therapies, understand when to continue taking them at home and or when to reach out to the practices. Next slide. We are looking at how we address side effects and toxicities. That's very common in everyday practice. Our approach, though, is more proactive in that patients will respond, we'll have outreach, and we're looking at other factors, particularly important for OAA therapy, which is patients' self-report of their medication adherence and their confidence. So changes really quickly. I talked about improving the rationale and some of the things we're looking at. We also plan to simplify the intervention. So we'll have a central process where we process the pros and send the report to the practice and provide the guidance on when outreach needs to occur. We've simplified the timing of it a bit where the patients complete the pro at week 2, month 1, month 2, and month 3 of therapy. And another comment related to feedback was really adding a cost analysis, looking at resources needed to conduct this trial. Next slide. So I think we've talked a bit about the benefit of this and hopefully will garner some interest. The next slide is really what we're hoping to gain, if you can go from you, is so we want to hear feedback that you have if you are interested. We received comments just inquiring if programming like this was even of interest to the sites. And so if you have interest, it's not a contract set in stone, but we'd love to hear from you if you have interest about participating or any questions so that we can make sure we include that in our resubmission. And we plan to resubmit in February with the changes we described. So I'm happy to take any questions, comments, concerns you see about what we presented.
So I think in the interest of time, we'll have to move on to, but I think what is really, really important for the team is if you do have interest, if you can email them, and we can also help provide everybody with their email contact, because it does help on these submissions to say, like, you know, we already have 25 sites that have expressed interest, and it's not a commitment, but they can then put it in as a list of sites that have heard about it and are willing to at least consider opening it. It helps them a lot. So we'll probably send out a survey for that, but any other additional comments, please just email the team directly.
Thank you.
Great. Just to keep things moving, we're going to have Dr. Christine Veenstra come up and talk about her study on colon cancer surveillance.
Thank you very much. It's nice to see everybody today. I know we're running short on time, so I'll be brief. So this is our current Together After Cancer pilot study. We've been pilot testing our dyadic module at our cancer center. Our recruitment goal was 10 to 12 dyads, completing both the module and the follow-up survey. We had an IRB amendment approved in April to expand our eligibility criteria because recruitment was slower than we had hoped for. And fortunately, that helped. So we have reached a goal. We've had 10 patient and partner dyads both do the online intervention module and complete the follow-up survey. We asked some questions about acceptability and usability. We got good feedback from respondents. They thought it was easy to use. It was just the right amount of time. It was helpful during their transition to follow-up care after active treatment for colorectal cancer. They felt that it helped both patients and partners communicate together about colorectal cancer follow-up care. One of our goals was to have patients and partners have a conversation about follow-up care after they had completed the module, and most of them said that they had done that, and most would recommend this intervention to others. So what are we doing now? We are working on developing a prototype of the clinician-facing piece of this intervention. So using information that we gather directly from the dyads, we will then give tailored information to clinicians about who the partner is, how would the patient and partner like for that partner to be involved, what are a few very simple tips that the clinician can follow to help achieve the kind of engagement that that dyad would like. We're working on a proposal for external funding from NCI. We're targeting a U34, which is a cancer prevention and control clinical trials planning grant, thinking about either a February or June 2023 submission. And our goals for this study, so our pilot testing has all been with spouses or partners, but we'd like to now expand upon this to be able to include non-spouse or other partner caregivers. We are going to work with clinicians across hopefully some diverse practices to identify the best way to deliver this module. Can we integrate it into the electronic health system, health record? Does it need to be a separate digital dashboard? Is maybe a printed piece of paper the best way to do this in clinic? We have primary and secondary study endpoints that we have identified as important, but we'd like to engage with stakeholders, patients, caregivers, clinicians, practices about what endpoints would be most meaningful to them. And then we'd like to conduct a feasibility study in 3 to 5 SWOG practice sites. And the goal here would be to develop preliminary data to support an R01 proposal to the NCI to support funding for a larger randomized controlled trial within the NCORE. And I'll stop there. Thank you.
Fantastic. Thank you. So if anybody has any suggestions or ideas or, comments, feasibility, I think your idea to expand the definition of caregiver from what we've learned is probably a good one, for sure. And we are excited about this U34 mechanism, and we'll work with the cancer care delivery team at the NCI as we develop that. Yeah.
Thank you.
So we just have a couple minutes left, and I don't know if Dr. Holcomb or Ocado is who's presenting. One option is we could just postpone this to the next time unless there's something you need immediate feedback on.
But you're welcome
Or,
to present.
yeah, you can present now very quickly, whatever is best for you.
Hi, this is Izumi. I think I can present very quickly on the updates on the study.
Okay, great.
So thank you for the opportunity to present updates on the study. Next slide, please. I'll be presenting on behalf of Dr. Holcomb. He's also online, and we appreciate funding from the whole foundation. Next slide, please. Briefly, the study focuses on improving rural patients' perceptions of care coordination, as there are many challenges with care coordination in rural areas. Next slide, please. And the study has 2 aims. The first is to assess early stage rural patients' perceptions of care coordination. And the aim 2 is to develop and pilot test a patient video education intervention that's designed specifically for rural patients. Next slide, please. And the study design, the first aim is a cross-sectional survey with 60 adult patients. And the second aim is a feasibility study with a single arm pre-post intervention design. And next slide, please. And the educational intervention is a TED Talk style patient video education that has 3 modules: cancer basics, care coordination, and how to be a self-advocate. And the videos are preloaded on an Amazon Fire tablet, so internet access is not required. Next slide, please. And the updates on both aims. The first aim, we are now working on data analysis for this aim. And the aim 2, we've recently completed enrolling 20 participants, which means we met 100% of target accrual. And for the majority of participants, they're currently working on the intervention. Next slide, please. And the next steps, aim 1, we are currently working on data analysis with the plans to complete by the end of this year. And aim 2, all participants, we are planning to complete study activities by the end of this year and start working on data analysis early next year with the plan to submit an abstract to the ASCO annual meeting. We also have plans to conduct a parallel study in Vermont with a larger sample size, and also planning on our grant proposal for a larger multi-site study in the future. Next slide, please. Thank you, Dr. Hirschman, Dr. Ramsey.
And you can see Izumi's contact information here, and feel free to send her comments or thoughts after the meeting.
Yeah, I think our goal, and we're super looking forward to your results. You can present them at the next meeting because it'll be good timing, and really trying to figure out how to transform these findings into a SWOG-led intervention. And then last but not least, I think we wanted to give Barbara, our amazing patient advocate for the Cancer Care Delivery Committee, a few minutes to give us updates from the patient advocates perspective and the Advocates Committee.
So I have talked a little bit today, but anyways, thank you for having me. I just completed in June my first term as a patient advocate, and Donna and Scott were kind enough to renew my second term, so happy to be here. The first 5 years is just learning. So please, if you have anything, an idea or things you want to run by me, you can write to me, we can have a call or anything. From the patient advocates, we have been in the last years recruiting community advocates that for any study that you do, we have veterans that now there's a veteran committee, but we had him as a community patient advocate for a couple of years now. And we have adolescent and young adults representation, LGBTQ, Latinx, and older adults. So if any study relates to that population, you can contact me and they can participate and help in the ideas. As advocates, we are also trying to help you guys, the researchers and the studies, to have more diversity and inclusion. So we're working on different ideas, and that's why we started these community advocates. And usually when we do our thought is when we do have an idea or gonna implement a study, from the beginning to see, who have the burden of the disease, how can we represent, and what are the sites that can really provide that population, right? Instead of trying to recruit everything, to be intentional for that. That's, the most important thing. I know we all have a tight schedule, so always happy to help in any way. Thank you.
Thank you, Barbara. And, as I mentioned, the patient advocates were so helpful for Tracer. They've been helpful for all the other studies. Please use this amazing resource as you develop your studies. And with that, we'll conclude the meeting, and thank you for coming, and hope you have a great rest of the SWOG meeting.